So yeah, thank you. This talk will be probably a little bit different to the sort of talks that I think you've been getting within this AViD Open Science Forum, in that this is not basic research, this is not preclinical, but instead going along the pathway to the development of these drugs into late stage clinical development. So what we would typically term phase 3 development, where we demonstrate the efficacy of drugs, but then actually going even beyond that to thinking about post-approval and how we still seek out to develop more evidence around these drugs post-approval, but also thinking about what the needs are for both seasonal influenza, which of course, as the name suggests, occurs every single year, but how we also prepare better for pandemic influenza. So hopefully some of these learnings you can take away, but very appreciative that probably this is a slightly different space to what many of you are thinking about. So hopefully it's useful. So as Alpha mentioned, I'm an employee of Roche, so a large pharmaceutical company who has been developing flu antivirals for a number of years now. I joined Roche about 5 years ago from a role that was somewhat similar to probably what many of you do, academic research, but also working within the WHO surveillance system within influenza. So let me just sort of level set everyone a little bit. Many of you are probably familiar with some of the flu antivirals, but maybe not so familiar with their history. So in the 1960s, actually, amantadine was one of the first drugs that was ever used for the treatment of influenza A. It wasn't specifically designed around influenza A, but it just so happened to have activity against that virus. Incidentally, not active against influenza B viruses. And then another molecule, rimantadine, was developed later in the 1990s, had improved or fewer side effects than amantadine, so it was taken on in that respect. But this class of drugs, the M2 inhibitors that bind the ion channel of the virus, were the first class of flu antivirals to really be used. Interestingly, though, as a result of widespread resistance amongst circulating influenza A viruses, both within the H3 subtype and the pandemic H1N1 subtype, these drugs are no longer active. A single amino acid substitution in the ion channel renders both of these drugs inactive. However, that's not the case necessarily for some of the avian influenza viruses. And so this class of drugs, although old, although hardly used anymore, should not be completely ignored potentially from a pandemic perspective as part of a pandemic response, but we can sort of put those to the side somewhat. In the late 90s, the first specifically designed flu drugs were developed, and this was after gaining the crystal structure of the neuraminidase, one of the glycoproteins on the surface of the virus. And zanamivir actually was the first of these drugs to be developed, followed very closely by oseltamivir, which was a structure that was a tweak on that of zanamivir, but making it more bioavailable, which meant that you could receive this drug orally. Both of these drugs are delivered twice daily for 5 days for both treatment and prophylaxis. And then a couple of other neuraminidase inhibitors, again, they're all targeting this very conserved part of the neuraminidase, peramivir and laninamivir, were also developed. And these drugs, and particularly oseltamivir, or the trade name Tamiflu, has been the sort of stalwart for influenza antiviral treatment for many years now. And really, you know, again, one of these lessons around drug development being simplicity of delivery, zanamivir arguably from a resistance profile was probably a better drug design than oseltamivir, but that simple difference between being a an inhaled delivery versus an oral delivery was the difference between one of these drugs becoming a blockbuster and becoming standard of care, and the other one really being hardly used. Lenanemavir, that bottom one on that list of neuraminidase inhibitors, incidentally, although it is an inhaled drug, it's a single dose, and so that has some convenience advantages. And in fact, lenanemavir has been the main flu antiviral used in Japan, the market leader for a number of years now. And as I'll touch on later, Japan is one of the few countries that really actively use flu antivirals for seasonal influenza. The polymerase inhibitors, and of course, as you know, the polymerase complex is made up of three subunits. Favipiravir, which is targeting PB1 and its role in replication. Quite a broad-acting molecule was also evaluated for COVID because of that breadth. Isn't really fully licensed for influenza and not really very much used, but I popped it there. The Japanese government have included or actually licensed favipiravir for pandemic use only, and only in the event that resistance occurs to all of the other antivirals that they have on hand. So I would put it in very much the bucket of an emergency use only. But baloxavir is a newer drug that has been licensed now in many countries around the world, and was developed both in partnership with Roche, where I've been part of this team, but also Shionogi, a Japanese pharmaceutical company who actually were the ones who designed the molecule originally. This is an oral drug and it's a single dose. So from a convenience perspective, it's the most convenient out of the lot of these. And we'll touch a little bit on the development of baloxavir and how that compared to oseltamivir and some of the lessons learned from that. So when we think about clinical development for seasonal influenza, actually there's a very traditional endpoint that has typically been used to evaluate whether these drugs work, and it's time to alleviation of symptoms. And this is typically around the treatment endpoint rather than a post-exposure prophylaxis endpoint, which is looking at when there is an index case within your household and we deliver the drug to the contacts of those index cases, what's the likelihood of them becoming infected, or what is the reduction in the likelihood of infection in those households. So treatment and prophylaxis are both typically evaluated when these drugs are developed through to try and receive marketing approval. Now, many of you would be familiar with what a viral curve looks like for influenza, probably in relation also to symptoms, but it's worth just reminding ourselves of the way that once viral replication starts, symptoms are delayed by a day or two, and then ultimately symptoms will peak and then tail off, and viral load will also tail off. But what we need to think about is when we've got an antiviral like baloxavir or oseltamivir, patients are only going to be presenting at a point when they have developed symptoms, particularly under a treatment type scenario, because of course they don't know they're infected until they start feeling bad. And often at the start of that period of time, they're still wondering, well, is this just a common cold? Is this just, the result of a bad night that I had last night on too many beers, or whatever it might be? People don't act immediately when they start feeling like they've got a headache, a sore throat, a bit of a fever. And so what you can see here that clearly if we're trying to shift this viral load curve, which of course is what these antivirals are doing, which is the black curve there, if we want to impact that, then we've got to catch people in this earliest window as possible in terms of symptom onset. And it's why this treatment window, and it's the same for baloxavir as it is for oseltamivir, in fact, all of the flu antivirals that have been evaluated, the treatment window is about 48 hours post-symptom onset. You wait longer than 48 hours, the viral load curve has probably peaked, it's tailing off. The impact of that antiviral on the various downstream endpoints, whether it be time to alleviation of symptoms or other things, is lost. Similarly, the earlier you catch it in that window, the greater the benefit you can see. So in places, and I'll use Japan probably in a few of those situations, or a few examples, Japan really has a culture of presenting very quickly to physicians. There is always an opportunity to get an appointment. There is a lot of rapid tests being used in physicians' offices. So what you have the advantage there is often you can catch patients within the first 24 hours of symptom onset, and that has the added benefit. Many of you would be aware that the efficacy of either oseltamivir or baloxavir compared to placebo in terms of reducing that time to alleviation of symptoms is about 24 hours. However, and that was the case for both of these drugs compared to placebo. However, what's interesting is when we look at something like baloxavir, which is a far more potent antiviral than oseltamivir, and you see that on the left-hand curve, that within 24 hours of treatment with baloxavir, you've knocked out nearly, well, over 4 logs of virus, versus oseltamivir, where you've only knocked out about 2.5 logs of virus, and you've still got some virus remaining. We don't have a placebo curve on here, but the placebo curve would be sitting even higher than oseltamivir. But for comparison purposes, you can see a difference in virological potency between baloxavir and oseltamivir, which may make you think, great, well, baloxavir is the way to go then when it comes to symptom reduction. When you look over to the right-hand side, this is the same bunch of patients who have had improved viral load reduction, but actually no difference when it comes to symptom alleviation. And this was one of the learnings, I guess, of some of the development of baloxavir, is even though this viral potency was there, there's enough going on in terms of cytokine production. We've seen enough of a viral load curve that some of those symptoms still aren't being alleviated any quicker, even though we're knocking out the virus faster. And this really, again, touches on this element of what we call patient journey. How does a patient get to treatment quicker? Is it through at-home testing? Is it through the role of pharmacies to maybe do testing or have stock on hand? Is it home delivery of antivirals? All of these aspects can probably make a difference. But apart from symptom alleviation, which arguably is important when we all want to get back to our life faster, there are endpoints which many would argue actually are far more important to understand and to impact the burden of influenza. And many of these endpoints, although are, in my view, very important to address, they're actually very difficult to answer through a typical phase 3 study. And the reason for this is the size and the complexity of many of them. What I'll do is I'll touch on a few of these endpoints or elements that ideally we would love to know the answers to for these antivirals. Many of them we're gaining, but we're not doing them all through phase 3 trials. So the first one is high-risk patients. So many would say, you know what, flu, yes, it's a burden for individuals who are otherwise fit and healthy. Yes, it knocks us out for a few days, but you know what, they're fine. It's actually the high-risk patients that we need to worry about, because if they acquire influenza, then there's a likelihood that they might end up in hospital. That's bad for them as an individual because they may go downhill. It's bad for the health system because that's a burden. It fills another bed, and there are resources involved. Then, of course, if you're hospitalized for influenza, then we want to know what the effect of antivirals are there too. What's the likelihood that treating a patient with an antiviral in hospital reduces their duration in hospital? How quickly will they improve? Will it get them out of the ICU potentially quicker? And of course, the most ultimate endpoint is how does it change the likelihood of death, which we know death is an outcome for many or some high-risk patients infected with flu. There's a third one which we're also really interested in investigating, and I think this is probably much more relevant now post-COVID-19, is what's the impact of antiviral treatment on onward transmission of the virus? What's the effect of knocking out that viral load in terms of reducing the chances that those around you, who live with you, your loved ones, your roommates, whatever, will get infected? And then, of course, the last one, when we think about pandemics, is, yeah, this is all fine, gathering this data about seasonal influenza, but how well are these things going to work when it's an H5N1 infection or whether it's H7N9 infection? So these are all questions that we ask ourselves and that the community ask as well, that are not easy to answer. But let's have a look at how we're trying to nail this and where the challenges are. So the first one, this high-risk outpatients, this understanding of the reduction in the likelihood of hospitalization. One of the realities is that the rate of hospitalization associated with influenza in the broad population is around 40 per 100,000 patients. But in patients who are a little bit, or in patients who are higher risk, and we take, for instance, those over 65 years of age here, which is one of the risk factors for severe influenza, that rate increases again. But nevertheless, to run a phase 3 study where you're trying to understand these events of, does this antiviral prevent you ending up in hospital, when they're so rare anyway, that means you're having to treat a hell of a lot of people to be able to find a handful of individuals who are ultimately ending up in hospital in the placebo arm, let's say, or in the comparator antiviral arm versus in the antiviral arm. In other words, studies that are either too large, would last for far too long, or would cost an inordinate amount of money that companies such as the one I work for would be hard-pressed to support it. So how else can we tackle it? Well, one approach is more pragmatic platform-type studies, and we can go into those in a little minute. What we realize, though, is that there are some scenarios where gathering this data is possible in a phase 3 type study size. And this is where the event rates are high enough where you can actually really measure this. And one example was, of course, during the COVID-19 pandemic, at a period where many high-risk individuals were very vulnerable to hospitalization. This was in the pre-Omicron phase and in unvaccinated patients. And the frequency of hospitalization was so high that we could actually, in the early days of the studies for things like Paxlovid, were able to get at this particular endpoint. What's interesting is for many of the COVID drugs that were developed a bit later and started their phase 3 trials in the Omicron period, where they had populations who were mostly vaccinated, many of those trials really struggled to recruit and really struggled to actually find significant endpoints because the frequency of these events was really so low. So we attempted to run we didn't attempt to, we ran, or Russia ran a study in hospitalized patients. Now we're shifting to this other endpoint. What's the impact on hospitalized patients? From our perspective, oseltamivir has become the standard of care in many hospitals. And it was unethical to run a placebo-controlled trial in a hospitalized setting, given that this standard of care, oseltamivir, existed already. So our study was to say, okay, well, maybe baloxavir plus oseltamivir compared to oseltamivir alone may result in improved clinical outcomes, shorter time for clinical improvement, reduction in the duration in hospital, the sort of endpoints that we're interested in. Now, again, interestingly, although this study where we combined 2 antivirals, baloxavir and oseltamivir, knocked out viral shedding within 24 hours. Again, a figure similar to what we've seen in outpatients, compared to essentially oseltamivir alone, which was more like 63 hours. Although that difference in viral shedding was present between the two groups, again, we didn't see any difference in that primary endpoint in terms of adding clinical benefit in these patients. Again, suggesting that the virus has probably done its job by then. Yes, we can impact the virus, but there is other things going on in these patients where knocking out that virus that little bit quicker is still not improving their outcomes. So it sends your mind down the path of, okay, well, are we thinking about immunomodulators here? We know that things like corticosteroids have been useful in COVID-19. How might that play into things? The other way that I think we can try and approach this, and we're supporting two large platform studies now, Recovery and REMAP-CAP. Recovery is a UK-based large network of hospitals, all part of this large platform trials. REMAP-CAP is more global, but both of them have the capacity to be able to evaluate a large number of different interventions at a very large scale, where we're talking many, many thousands of patients. And ultimately, these are adaptable trials where the blinded data feeds back into the algorithm of how patients are randomized, such that the interventions that are looking more encouraging are more randomized more commonly to patients, allowing those interventions to be able to be greater evidence to be generated, and those that are showing a lack of efficacy are identified relatively early. So this is a really exciting element, and I think data from both Recovery and REMAP-CAP that will probably come in the next few years should give some really insightful elements around the role of these drugs. Other endpoints are also important, but can be very hard to capture. So transmission reduction, for instance, is one that, as a result of our viral load impact that we're seeing with baloxavir, we asked ourselves the question, well, okay, well, if you're no longer shedding virus, surely that should mean that the likelihood that you're transmitting it to somebody should also be reduced. Now, we're used to thinking about prophylaxis as a way of reducing transmission, where you give the drug to the uninfected household contacts. But this is a slightly different way of looking at it. And so we designed this trial that started back in 2019, and we are soon to finish in the next month or so. And index patients who were infected with influenza were randomized to receive either baloxavir or placebo. They had to be within their 48 hours of symptom onset. And then you also enrolled their household contacts. And it's actually ultimately these household contacts that we're most interested in following up, because what we're doing there is swabbing them on many days after enrollment. And we're ultimately asking the question of, do those household contacts become infected? With the primary objective of understanding what the efficacy of baloxavir is in terms of reducing this onward spread. So that's some exciting data that would probably change a little bit how we think about flu antivirals, where right now we think only about the patient getting better faster, avoiding complications. But now we may be also thinking about what does that mean at a population level or community level in terms of reducing onward spread. The last endpoints that I think are important to capture that are quite tricky, particularly from a pandemic sense, is how will these drugs work in a future pandemic? Now, of course, we can gather in vitro data and we can demonstrate that IC50s are comparable, whether it's for an H7N9 virus, H5N1, or a seasonal H3 or H1. But I think ultimately, and we can do this also with animal models, but I think we really only have one insight, and that's from the 2009 pandemic of how some of these drugs performed against an influenza virus that was somewhat novel. And I say somewhat, of course, because this was an H1N1 pandemic and we had seasonal H1N1 circulating before. But even then, there is some really nice data from very large meta-analyses for oseltamivir showing that neuraminidase inhibitor treatment really did substantially reduce the mortality risk in patients. Very large studies, 29,000 patients. So again, it's demonstrating the challenges of gathering this data, and of course, trying to answer these questions ahead of time for a future pandemic, which we don't know which virus will be causing that, is of course very hard. So when we think about seasonal use and pandemic use, clearly we know that these antivirals target conserved parts of the virus and so have broad activity. So we don't need to update them, unlike vaccines. But the challenge is that unlike vaccines, which are very broadly used around the world, flu antivirals typically aren't used in all that many countries around the world at a seasonal basis. In fact, there's only a handful, and it's mainly Japan, US, China, and Russia, who typically have active use of flu antivirals for seasonal influenza. But the big challenge that I often talk to the organization I work for, and as well with WHO and others, is when we've got a key tool to use in a pandemic, like influenza antivirals, that has a certain usage for seasonal influenza, how do we get it to the point when a pandemic comes that we will have sufficient supply to be able to utilize in a pandemic? The big challenge is that the manufacturing timelines for many of these drugs is very long, 18 to 24 months at best. And maybe you could argue that in a pandemic, it may be even tougher, because the supply chain that is typically used for these moves different parts of the product along the pathway of manufacturing all around the world. We know that things can hit hurdles in a pandemic in terms of shipping and movement of goods. So we've got these very long timeframes to start producing these drugs. And so what it means is that we are not in a position that when a pandemic comes, we start saying, right, let's start making some more of this, because 24 months later, that's when you're going to see the fruits of your labor, and that's not very helpful. So how do countries and how do organizations manage that? Well, for high-income countries, they approach it via stockpiling. But this comes with pain points, and stockpiling is where they would purchase a large amount of antivirals. Typically, the sort of percentage of coverage is thought of about 20 to 30% of the population, such as in the US. But it comes with pain points. These drugs get put into a warehouse. They slowly expire. They cost money. If the pandemic doesn't occur, that's considered a bit of a waste. So there are challenges that come even for high-income countries. And so there are some thoughts about, okay, how can we approach this? It doesn't make sense when many European countries, for example, have pandemic stockpiles. The drugs sit in warehouses. Seasonal influenza comes through every year, but very little antiviral usage is there. So is there an opportunity to rotate some of that material out of these stockpiles, getting the benefit of treatment of seasonal influenza, and then restock so that your pandemic emergency stock is there? But that's all well and good for high-income countries. But what about low and middle-income countries who clearly have got much smaller health budgets and can't afford, in many cases, to be able to stockpile? So there are approaches. There are WHO donations that organizations like Roche, and who I work for, commit to in terms of provision of antivirals in the event of a pandemic. And there are other initiatives. The PIP framework, which many of you may be aware of, is another initiative that involves industry contributions to help with this. But there are other ways, voluntary licensure initiatives. An organization that's relatively new called the Medicines Patent Pool works with companies like Roche to say, okay, well, we will work with you and a generic manufacturer to provide the license to make this drug available in certain territories. The challenge is, of course, is making that work at a scale that's meaningful for those regions that it's looking after. So this element, and of course industry is a player in this, how do we also improve our investment and our ability to scale up to seasonal demand? And there are some manufacturing approaches as well as simplifying supply chain that could all help with that. So to bring all of this together, hopefully I've given you a slightly different snapshot than you're used to in this space of what happens at the later stage once these drugs are in late stage development or have achieved licensure. I think there's been success in developing flu antivirals that are safe and effective, but their broad use in general has been limited to a handful of countries that have health systems that really value the way that they work. But it does mean that there is this strong supply gap between what we can do for seasonal influenza and then ramp up to when all of the world says, hold on a minute, we need these drugs in a pandemic. I think there are clear evidence gap in key patient populations that have been hard to address via phase 3 trials, and so traditionally have been sort of ignored. But I think now with large platform trials, we have the ability to try and be able to answer some of these questions. And this helps us understand the appropriateness of their use in various different circumstances. I think I've shown you probably an example of where improving virological potency doesn't always translate into improved clinical endpoints. And inherent in that is this patient journey, the way that we manage patients when they present, how we find ways to try and achieve treatment earlier and therefore see the efficacy that is potentially there for these drugs, but often is lost if patients are presenting late. And then there are these inherent manufacturing and supply constraints that mean additional investment is really needed in this area to be adequately responding to a moderate or severe pandemic. I hear far too often the expectation that in a pandemic, it's okay, we'll just turn on the pill machine and everything will be fine. And I wish it was that simple. And I think one of my learnings in going from more academic and public health research into industry is to try and connect those two worlds in the realization that it's not quite that simple, but what do we do to try and solve this ahead of a pandemic? So that, I think, comes to the end of my talk. So let me stop sharing.